Thanks, Yuhui, for joining. For those who don't know Yuhui, he is a research scientist in Illinois, University Illinois of Urbana-Champaign. He's mostly famous for his work, in my circles, he's mostly famous for his work on vapor, this sequence contrast optimized for layer-dependent fMRI that combines CBF and CBV to get rid of the veins independent of other contrasts. And today he will talk about mesoscale, brainstem-prefrontal-striatal pathways underlying salient decisions using high-resolution fMRI. Please, Yuhui.
Yeah. Uh, Thank you, Renzo, for the introduction and also the invitation to share this work. So this work was done in collaboration with Josh Gao, who was a professor from Taiwan. So he did a lot of like decision-making work, and he visited us like for a summer a few years ago. So we did this collaboration using 70 high-resolution fMRI to study the mechanism underlying the decision-making. And through this talk, like in this community, like everyone's interested in Leo specific fMRI. So after this talk, I hope like to give you introduce some. So through this work, I hope you have a feeling like high resolution fMRI is not only useful for Leo specific or column specific in cerebral cortex, but it could also be very useful to study the function in those very small microstructure in subcortical areas like brainstem, striatal. So I will start with some background of decision making. So decision making is a fundamental cognitive process that guide everyday behavior from simple choice to complex life decisions. And in this talk, in this study, we are going to focus on the value-based decision making, and you can understand this value-based decision making processing with like several stages. So the first stage is the evaluation. Can you see my cursor moving? Like, great, great, great. So the first stage is evaluation. Like think about it, you have like a gambling task like this one, and you have a half chance to win thousands of dollars, and also you have half chance to lose. So the first thing your brain is going to process this task. You will evaluate the risk reward trade-off, right? Uses some of your brain areas. So after evaluation, we need to make a decision, do a choice commitment or action selection. You either accept this gambling task or reject it. So you to use based on like some brain regions. And after the action selection, there will be outcome processing stages. So there will be an outcome. If you accept this task, there will be either like you will win the money or lose the money. But if you reject it, there will be nothing changes. So there will be an outcome. So basically these stages, evaluation, choice commitment, outcome processing, and which different stage will be processed by different brain regions. So the central question here is how do the brain coordinate this evaluation, action commitment, outcome processing within one decision making? And there have been many previous human fMRI studies have identified major cortical and striatal nodes involved in this value-based decision making. It's mainly the prefrontal striatal pathway underlying this decision making. So in prefrontal cortex, like ventral prefrontal, DLPFC, they will be involved. And also in striatal, there will be like a caudate, putamen, and also nucleus accumbens be involved. If you understand those different regions, different nodes based on the different stages, so in the motivational evaluation, like prefrontal cortex, caudate will be involved processing this motivational evaluation. And for the action selection, putamen will be responsible to make the choice, do the action selection. And for the outcome signal, outcome like either rewarding gain or loss, like nucleus accumbens, also VTA, which is free, is a very well known like rewarding processing like a nucleus. And also prefrontal cortex will be involved in outcome processing. So those studies basically have identified where decision-related signal appear, but it doesn't explain how the information is dynamically gated. So how the signal is gated into the pathway and also transformed between different stages, between different nodes, and how they are organized across the pathway during a single decision. So in this study, we are going to understand those hidden mechanisms underlying this pathway. And we think a lot of those mechanisms are at the mesoscale level. So we want to design decision-making conditions in which this pathway-wide computation are most widely engaged. Because we think a lot of mechanism underlying this pathway is mesoscale. They are weak, they are very weak signal, and in some very small subcortical structures. So the signal are really weak, so we want to maximize the response so they can be detected by fMRI. So our strategy is to focus on the silent decision. So what's a silent decision? It's basically most of the decisions are routine. They are just a regular state. Doesn't make a big difference. But some decisions are highly consequential. They are, for example, unusually important, make a huge difference, and require rapid commitment and uncertainty. For example, like some super high stake life decision, emergency response, or some financial investment, which means you could either win a lot of money or lose a lot of money. So for those super high stake decisions, we are going to focus on this study. And we think for those super high stake decision, or we call the silent decision, it requires neural mechanism supporting decision that could be amplified under those conditions so we can better study it. And for the neural mechanism supporting the silent decision, one potential mechanism is the LC-mediated silence gating. So what's the LC? LC here, so we mean the local serratus. It's a tiny brainstem region in here like about 2 millimeter wide. So it's a very small region, but it's extremely important because it's the brain's primary source of releasing NE. And it's also a key region, like one of the earliest brain regions that can be detected, for example, during age development or during some like a neurodegenerative disease like Alzheimer's. So it's a very small region, tiny region, but it's very important. And its activity increased during silent or unexpected events. So we think during like a silent decision, this region will be strongly involved, acting as a gate into the decision-making pathway, and it can globally increase the neural gains through distributed brain regions. Also include for the silent decision, basically it can modulate, amplify the decision-making pathway. But this region, so given this like those knowledge, we kind of hypothesis LC it will serve as a silence gate, as a gate to like amplify gate into this pathway, but also amplify the coordinated activity across the decision-making pathway. But this region is also quite difficult to study because it is in a deep brain, in a tiny brainstem nucleus is very small. And it responds to the silent decision, silent events. So silent event, it cannot be regular, cannot be repeatedly. So it has to be real and unpredictable. So it's quite difficult to design a task to reproduce this response like in an experiment. So we need to design a task that capable of reliably induce these silent decision states. So I will introduce in next few slides. So this is the first question we want to answer during this for those decision making, like one mechanism we want to like study. And the second question we want to answer in this study is like how is valuation transformed into action. So as I introduced in first slide, decision making you understand it as different stages, for example, from evaluation to the action selection, and then there will be outcome processing. So the question is here is how the different stage they are transformed from one stage to another. So here how is the stages of evaluation transformed into the action selection. And we think we are going to focus on one candidate structure. We call the name is like green mat bridge. And the green matter bridge, for those you may not be familiar with, so green matter bridge is like here we are referring to the candid. So like in stratum, like in the stratum, here is the stratum. There's like here is this is the caudate, this is the putamen. So between caudate and the putamen, like so during the development, like when the baby was just born, like caudate putamen, they initially like was developed as a continuous like structure. But with development, they become like separated by glowing internal capsule, which is white matter fibers. But still, like so, those two like structure will gradually like separated. But still, there's some leaving like a residual green matter bridge connecting those two structure. So you can see here's the caudate, here's the putamen. In the human adult, you can see a lot of like those kind of residual gray matter bridge connecting those two structure. Also in the T2, like so here is the MT weighted EPI, the high resolution, we can see this structure. And also in T2 weighted image like 0.8, you can see caudate, putamen, there are some like a residual gray matter bridge connecting those two structure. And in the here is the FA map from diffusion. You see there are some like white matter fibers interleaved with gray matter bridge. So there will be some gray matter bridge connecting those two. So this gray matter bridge it contains like a neural cell body rather than white matter. It's gray matter. Like people always when people detect some activity in those areas, maybe you think it's white matter, but it's gray matter. If you have like sufficient resolution, and it's also very thin. It's about 1 millimeter wide in human adults, and it provides structural continuity between caudate and putamen, and based on the previous decision making, those kind of knowledge, we know like caudate is a center processing the value evaluation, motivational evaluation center, and for the putamen, it is mainly responsible for the action selection. So so here this gray matter bridge is basically located at an interface between the evaluation and the action selection. So based on this unique position, we hypothesize this gray matter bridge it acts as a law, like a facilitated transition from variation to action. So this is our second hypothesis. And but you can like imagine the challenge to study this gray matter bridge. First, it's very thin. It's 1 millimeter like a thin bridge. So you need a sufficient resolution, and also previous basically I didn't find any previous literature like studying the function of this region, and when we like start first like initiate this study, we don't know if the gray matter like gray matter bridge like the response can be detected by fMRI. So this is our second hypothesis we want to study. And the third question we want to answer in this study is, which is more familiar by this group, is the layer specific function. So how are decisions organized across cortical layers? And what we know, like from previous studies, like a prefrontal cortex, it participates, it will be engaged by decision making by multiple stages. So it will be activated in choice selection of the decision making, and also it will process outcome from the decision making. So prefrontal cortex participate in multiple stages of decision making, and we know like different cortical layers they can support a very distinct computation, like in working memory, different stages, like different layers we involved in different stages. So here our key question is how different stages of decision making organized within the same prefrontal region or subregions. So our hypothesis choice selection, like a choice selection outcome signal different stages, will they be segregated across cortical layers within prefrontal like cortex or subregions? So this is our third questions we want to answer. And based on all those previous study knowledge and also our proposed hypothesis, we propose this, we come out this kind of like a mesoscale framework for the decision-making pathway. So we have this framework. So during this framework, LC, it acts as a, like a silence gating into the pathway. It could modulate the whole pathway decision-making pathway, and also even like at like a large brain scale level. And so this is the LC law. It acts as a gating and also could modulate the pathway. And the second law is the decision making in this pathway. Gray matter bridge. Gray matter bridge is located at the interface between the evaluation centered in cortex and also the action selection in the putamen. So we think a gray matter bridge could facilitate the transition from evaluation into action selection. And the prefrontal cortex layers, we think it could segregate different decision-making stages. And also with some additional piece from, we know from previous study, for example, VTA, substantial nigra, and also nuclear components, it will be responsible for the rewarding signal, and also already know like cortex responsible for the evaluation, putamen responsible for the action selection. So we propose this framework incorporate the previous knowledge and also some mesoscale mechanism we need to test. So this is our general picture. Like for this study, we want to answer those mainly three parts. I would say gray matter bridge and prefrontal layers, and so to study this mesoscale framework, we use a lottery choice task. So lottery choice, you can think it as like a gambling task. It's basically like for each trial, the subject was presented with, for example, like some like a lottery, some points, or like in the real world it will be money, but in the experiment it's like the points. So the goal of for the subject is to maximize. They want to accumulate as much points as they can during the experiment. So in each trial, it present like a stake, like for example here 61 points, and they have 78% to win and also 22% to lose in this example. So during a time window, they can either accept or reject. So they were offering a choice, they were offering a stake, so they could evaluate the reward magnitude and probability. And they also have a time window to do the action selection. Either accept or reject, this will be like a choice selection. So we call this phase as a choice selection phase. And after the choice selection, after a short time interval, there will be an outcome like after the decision making. So if they accept this task, they could either win the points or lose the points based on the stakes. And if they reject it, they will still see the results like the missing points, but nothing will change. So with this task, this task was like it's not new. It's already established in previous decision-making studies, and we adapted here. We used it for this study because it has a very clear like separation of different decision stages, like choice selection. In choice selection, there will be like evaluation, action selection, and in the outcome like a phase, there will be like a gain or loss reviewed by the decision. So we can study based on this task. We can study the transition between different stages, and also we can study how different stages will be processed by different cortical layers. And more importantly, and also one key change like we made in this study is we add silent decision conditions. So as we see, most decisions, they are routine, just the regular stake. For example, in this example, the regular stake we mean, so they have like a stake points of like less than 100, like a small points. So either win or lose, they just like doesn't make a big change. It's just less than 100 points. And also for the regular stake, the win probability could either be low, medium, or high, and also a regular like response window here 3 seconds. But at some random time points, we add like during the run, we add some like a few super high stake. So this is the key. Also, we are going to focus on the super high stake. We add some super high stake trials. So the super high stake trials basically it featured with extremely win or loss, high consequences. So compared to a regular stake, it only has like less than 100 points. For the super high stake, it has like thousands of points. So it could make a huge difference. The subject will win at some time point. They could either win a lot of points or lose even to negative. So to make a huge change, it's a high risk, high reward. And also it's very rare. We only present this like in about 40% of the trials, and it's randomly presented, so it's unpredictable. And also it's urgent. The time window for those super high stake is like a half. Like they need to make a choice selection within like a within a time when limit like a half of the regular stake. And also it is those super high stake they all under like some uncertainty. There will be a balanced win or loss. So they cannot really like be sure consistently lucky or unlucky is basically the balanced win or loss. So all combine all those features we design this super high stake choice to create like a silent decision conditions. And the purpose to add this is basically we can, based on this super high stake silent decision, it can recruit LC mediated silence gating. So we can activate this LC gating law and also to amplify the pathway-wide mesoscale computation, increase the detectability. Because a lot of mechanisms we want to focus in this study, like LC green matter bridge cortical layers, the signal change will be low, and in those, for example, LC green matter bridge, the structure itself is also very small. It's not a direct response, so the response is going to be very weak. So we use this super high stake condition to maximize the response so we can detect it by fMRI. So this is so far is our background and also task design. And for our acquisition, we use 70 high resolution fMRI. For function image, we use 3D EPI and 0.8 millimeter isotropic resolution, 18.6 slice covering this pathway, brainstem-prefrontal-striatal pathway with a TR of around 3 seconds. And we are using bold here because a lot of our targeted region here are in subcortical, like LC, striatum, green matter bridge, those kind of regions, VTA. So they are in subcortical. And also this task design is like is a event related design because you can see in the task, like for example, the time interval is like 3 seconds, 1 to 5 seconds. So we cannot really use more level specific, more spatial space specific like a contrast. So we are using bold here. But like for bold, it has superficial bias. So for the layer, for example, for the prefrontal cortex layer function, we do a normalization like of layer profile of super high stake by the regular stake. So we normalize like normalize for example super high stake by the regular stake layer profile to reduce this layer dependent bias to highlight the stake related modulation instead of the absolute signal change from one condition. So we take some like take this normalization to reduce this bias for layer analysis. And for the anatomical reference image, we are using the magnetization transfer weighted EPI and using an identical EPI readouts for both between function and anatomical image. So in this way, the function image and anatomical image they matched during acquisition. So we don't need to do any like alignment or registration. We can do all analysis, including like determine cortical depth or ROI in just based on the MT-EPI image. So in our native fMRI space, so for example, like here in the LC, so here like all the background underlay is the MT EPI imagery. So it also gives us a very nice contrast in the subcortical region, like for us to define, for example, gray matter bridge, LC, and also for the VTA, like here, the middle is the red nucleus, both sides is the substantial nigro, so the VTA is in between, and LC is MT. It has a like in previous study we know like MT is sensitive to neuromarine, which can be generate some contrast in LC, also in gray matter bridge. So this identical EPI acquisition between functional and anatomical image really helps us to do an accurate analysis in this subcortical region. And you can imagine for the gray matter bridge, every subject has a unique structure. And it's so thin, it's 1 millimeter. You cannot really do registration or have any kind of blurring. Otherwise, the signal is not specific or will be gone. So this is our acquisition. And if you have any question, please let me know. Otherwise, I will move to the results part.
Maybe I have a question. Like those about acquisition, those deep areas are kind of hard, right? At 70,
Yeah.
specifically with the Nova. So did you do kind of PTX specific things? Do you use the PTX coil in CP mode, or what's your RF hardware component here?
We do use like a PTX coil because in some subjects, for example, in those LC, is quite at the bottom of the brain. So for some subjects, if they have a larger brain, the PTX coil will definitely help to increase the imaging intensity in those regions. Yeah, but just use the PTX with a CP mode, no, nothing special.
Gotcha.
Yeah.
And you were happy with the TSNR values down there. Like I imagine you need to have like double digit acceleration here with like almost 90 slices in 3 seconds, so that
Yeah,
the
yeah.
chip factor might be severe.
Yeah, so we have 86. We didn't push it so the acquisition like here for the 3D API is not aggressive at all. It's 0.8, 86 slice, and the TR you see is not very short. It's 3 seconds, and we use a normal CAPI, like a skip CAPI, like a 3 by 2. So it's not aggressive at all. It's very regular. It's a very robust pad. Yeah.
Gotcha.
Mm.
And we have a question from David Feinberg.
Yeah.
How did you validate normalization of super biregular stake profiles for Wayne bias?
Uh, we saw some in some previous studies. They also did those kind of for so normalization to have reduce this superficial bias. We didn't do specific for example did some yeah did anything special for example to validate this. Yeah.
And by normalization, you mean kind of division of
Yeah.
the different betas, I guess.
Yes. So to focus on the state-related modulation instead of for any instead of a single condition.
So
Yeah.
you're kind of normalizing by the like CVR, by the vascular gain
Yeah.
that you have in each layer,
By baseline. By baseline. Yeah.
but you're not kind of deconvolving. You're not taking out of
No,
spatial
no,
misplacement. Okay.
not a deconforming. Yeah.
Okay.
Okay. So so in following slides, I will show you the results. So first is the super high stake modulation. So so
Wait, sorry. We did had a raised hand here in a second. Who was it?
Sorry, that was a
Okay.
mistake.
Oh, okay. Never mind. Sorry. Continue, Yuhui.
yeah. It's fine. So first, for basically for the whole pathway from brainstem to prefrontal to striatal, basically the whole pathway, all nodes, they could be very strongly activated by the super high stake decision first. So you can see in the LC here, like a very small activation cluster. Activation induced by super high stake, and also in the VTA substantial nigro here. And the prefrontal is very obvious. It's a very extensive activation. And in the stratum, you can see several separated activation cluster. In the caudate, which is responsible for the for the evaluation, and also for the in the putamen here is which is responsible for the action selection, and also NAC. You can see they are very clearly separated. In 3T, you may also see the activation is striatum, but it's not so clearly separated in the caudate, putamen, and NAC. And also you can see a very beautiful like activation just along a very thin like a bridge here in the green matter bridge between caudate and putamen. Also, we see it here. I will show you more examples to show how robust it can be detected. But basically, the whole pathway can be engaged, can be very strongly activated by the super high stake decision. And the right basically shows the signal change across different ROIs and comparing super high stake, which is in red, and with regular stake, which is in blue. And each dot is like one subject. So here we cannot do the alignment, do a group analysis, walks-wise group. Instead, we basically extract the ROI level, like a single change, and do the group analysis for these different ROIs. And so to understand these results, so basically we can put a line here. So before, like on the left of this line, the super high stake basically significantly increase the single change compared to the regular stake. But on the right side, there's nothing changing in green matter bridge, putamen, which are insensitive to the stake value. So what does that mean? Basically, on the left side, those LC, prefrontal, caudate, they are responsible for the motivational evaluation. VTA and nucleus accumbens, they are sensitive to the rewarding value, but during the feasible choice evaluation, motivational evaluation, you kind of have expect you are going to win or lose. So all those regions they will be sensitive to the stake value. But on the right side here in green matter bridge putamen, they are insensitive to the stake modulation, insensitive to the stake value, suggesting their low are more towards the downstream execution after evaluation. So those two, green matter bridge putamen we already know is responsible for the action selection. And for the green matter bridge, here we also like it basically suggests the green matter bridge law is also more similar to the putamen. It's more toward the execution after the evaluation is done. So we have some information here about the green matter bridge function. So this is the first. Second, if we take a difference here, so from previous slides, if we take a difference between different stakes and have those differences like contrast of super high minus regular stake, so you can plot this stake fact, basically the stake sensitivity of across different ROI. So we kind of find like LC it basically show the largest like those kind of super high stake enhancement among the whole pathway. Green matter bridge putamen, no, we already know they are not sensitive to the stake value. And LC show the largest like enhancement by the super high stake. It suggests it support LC as a silence gate into the pathway. And more evidence about the LC low. So this is the decision-making pathway only. Actually, this effect could be extend beyond the local decision-making pathway to a global brain like system level. So also compare the super high stake with regular stake. So in all those major network, in default model from the parietal limbic silence, super high stake, they basically enhance all the activity in all those major network because of the silence effect. And more interestingly, in default model network, we also saw a concurrent stronger suppression of the default model network. This kind of like I think this is kind of consistent with the default model network law, because during some task, if you have like increased like attention like a load or computation demand, default model network will be surprised. So we also see the default network be surprised to help the subject to redistributed computation results for a better decision-making behavior. And this is consistent with the LC mediated LC law acting as like a silence gating law to modulate the decision-making pathway, but also basically the whole global brain system acting as like a global like a modulator law, including both the decision-making pathway and the whole brain level. So this is about the LC mediated like silence gating and also modulation. This is the first part. So the results of the LC law. And the second is the decision is for the gray matter bridge. So for the gray matter bridge, in previous slides, I'll only show you one example. Here, so here is the map, gray matter bridge of different like subjects. So this is the first subject you see like activation along the gray matter bridge here. Second subject you see here. And third subject you see it here, and maybe also here, and also here. Fourth subject here, the other subject here, and the subject here. So you see this gray matter bridge, they are very consistently activated across different subjects during the super high stake choice. And regarding its law, regarding the function law of gray matter bridge, we did a fuller comparison between the different commitment. Sorry. So we compare. So like for the same evaluation condition, for example, during the same like a stake magnitude, for example, super high stake, so it'll be same evaluation. Because it has the same probability and uncertainty, so the evaluation will be same, but we compare the different response speed, so different commitment. So we compare the short versus long response time, like during a same evaluation stage, and compare those for all those RI. We find only gray matter bridge show a significantly great activity during rapid decision. So here, like here, gray matter bridge, like the blue basically means like a fast decision making. So gray matter bridge show like a high response to a faster response. So it is supporting a law in facilitating the transition of evaluation into action. So based on those two slides, this one here, gray matter bridge is not sensitive to the stake value. So its law is kind of toward the downstream execution after evaluation is done. So gray matter bridge definitely not sensitive to stake value. It's not related to the evaluation. And also this slide, it support a law in facilitating the transition of evaluation from evaluation into action. So based on those two results, basically we can understand like gray matter bridge function. It's after evaluation, and it acts as a law, facilitate transition from evaluation into action, toward action. Yeah. So this is the gray matter bridge like a part results. And for the outcome processing after the evaluation, action selection, the third stage is in the decision making is the outcome processing. So for the outcome processing, we know it will involve like nucleus accumbens, VTA substantia nigra, and also prefrontal cortex. But to differentiate these different outcome, for example, in the outcome there will be is a gain or loss. So here we show like a nucleus accumbens and also VTA, they can actually not only process the outcome signal, but they can also differentiate the gain versus loss. Is a win points or lose the points, it can like differentiate those two in those VTA and also nucleus accumbens. So this is consistent with their law because we know VTA nucleus accumbens is related to like a dopamine related like a rewarding signal. And the interesting thing here is prefrontal cortex. We know prefrontal is engaged in processing the outcome signal, but at a regional level or sub-regional level, we didn't see, for example, prefrontal could differentiate gain versus loss. It could process outcome signal, but couldn't differentiate gain versus loss. And we think here like gain loss encoding in prefrontal. Maybe it's at a finer, like a spatial scale, not at the regional level. But later I will show you, it will emerge, it will come out through a Leo analysis. So so for the Leo results in for the Leo results for the whole prefrontal cortex, we divide it into different subregions, like four subregions. But in two of them, in ventral prefrontal and also dorsal prefrontal, we see a significant interaction effect between different decision-making stages and cortical layers. Like here, for example, the blue curve show the laminar profile of the choice selection, and the red curve will show the laminar profile of outcome processing. You see, they basically those two stages they engage very different like laminar part. Like a choice related activity, like the blue mainly engage the deep layer, and the outcome related activity mainly engage the superficial layer. And those those interaction effect of between stage and cortical depths, it show a significant interaction effect. And we see this similar like laminar effect in both ventral prefrontal, which ventral prefrontal also include limbic system and also include some like those kind of quite bottom of the prefrontal, and also those DLPFC. And these Leo results actually could also show some like behavior dependent effects. Behavior dependent effect, as I mentioned in previous slides, like in this slide, prefrontal cortex it cannot differentiate gain from loss at a regional level. But actually in Leo, at a Leo level, we see these effects. So here, like a gain versus loss, we see like a laminar organization of the outcome rate response differed between these gain versus loss in both regions. So gain versus loss, it doesn't show, doesn't different, it doesn't differ at the regional level, but actually it show like a Leo effect. It mainly those kind of outcome valency effect is mainly in superficial layer. And for the actor first reject the different choice selection. It also shows some, both of them are toward like a deep layer. And in the DLPFC, we see a significant greater effect in deep layer, relative superficial for the accept first low, for first reject in the DLPFC. So here, this slide, basically those two slides showing you like a different decision-making stage, they could be segregated across cortical layers. And also these behavior related, for example, for the outcome, it has different behavior, for example, is gain or loss. Or for the choice selection, it has accept or reject those behavior actually could also be related, could also show it in the laminar effect. So that's the whole talk I want to share. To summarize it, basically we propose this pathway. And for first, for the LC, it acting as like a silence gating law. It could amplify this pathway-wide neural activity across like extensively, modulate extensively in the decision-making pathway. And also it could modulate even the global brain system to help redistribute those kind of brain computation results toward a better decision-making behavior. And for the gray matter bridge, we show very consistent activation across subjects. And its function previously is unknown, but after this study, we know gray matter bridge law is this law. It functions after evaluation. It facilitates the transition from evaluation to action selection for a rapid decision commitment. And for the prefrontal cortex layer, it can could segregate the choice selection and outcome processing, and also is related to the behaviors. And if you are interested in more details, you could like find it in this paper. And that's all for my talk. I'm happy to answer any question if you have. Thank you.
Thanks a lot, Yuhui. Wow, this is quite a lot. Thanks for walking us through this nice paper. And there are some beautiful activation maps and then the results there. We have a couple of questions. Maybe I go in order. Atana Akbari has a question. Do you want to unmute, Atana?
Yeah, thank you for the great talk. I just wanted to know if you have looked at the reproducibility between sessions of the same subject of the gray matter bridge. I mean, was it reproducible in a single subject, location-wise?
So each subject is just a scan once. We didn't so we don't have
So
like
you scanned each subject only once.
yeah, because for those tasks, if you scan in the second time, because it has those super high stake, it's kind of insense the behavior change.
Yeah.
You cannot
Okay.
repeat.
Hi, I think I'm next. So my question was about, in your introduction, you talked about how you were expecting to find outcome layer processing in superficial and choice in deep. What led you to hypothesize that?
Sorry, can you repeat your question?
It's about this exact question, yeah. So what led you to hypothesize that superficial would be more involved in outcome processing and deep in choice?
Okay, yeah, that's a great question. So like some motivation first like different layers they supporting different very distinct
Yeah,
motivation. And the second is actually we are also motivated by the DLPFC study, like by Renzo and Emily Finn, like so for working memory, like so superficial deep layer. So in for example in a working memory study, deep layer is also responsible for action like for action, I think after working memory, right? So yeah, so if and here is basically we initially we think this task decision making is actually working as a back out working memory. So working memory, you see something, you hold the memory, and you press the button. So the action is like at late. But here you do the action selection, you press the button, make a decision, and then you have the outcome. So it's kind of like a back out the working memory, and the lamina effect actually comes out exactly back out the working memory. So that's our initially like how we hypothesis this layer effect here.
thank you.
It's like an input-output layer model, is it?
Yeah. Input. So here, like a deep layer is responsible for the choice selection.
Right.
For the choice selection, you need to make an action selection, right? It's kind of output. That's how we.
I guess my thing is that the what's considered the input here would be the actual values, the actual stakes, but that's not part of your model. And I suppose you think that the superficial layers might be sensitive to the actual stakes.
So here, like for the stake value and the action selection, actually we cannot separate it. So the choice selection we consider as a same decision making phase, because during our task here, like those kind of choice phase, it include both evaluation and action selection. We cannot really separate because it's the time interval is like a few seconds. We cannot separate. But yeah, so that's why.
That makes sense. Yeah, thank you.
David?
Yeah, it's a really interesting talk. Thank you. And I'm wondering, correct me if I'm wrong, but you mainly are doing the layer analysis in the prefrontal cortex. The other regions are a little more difficult to get with the resolution. You're looking at also is that correct? I guess my real question is, given the concern of venous bias and your normalization method, couldn't you use in addition to what you did a different method just in the prefrontal cortex, like that's not sensitive to the venous bias, in addition to what you did to kind of validate or double show, like either vaso or grace or spin echo EPI, that would independently show the findings that you have? or is that not possible statistically?
Here we didn't do any additional like experiment just to focus on prefrontal to so we didn't do additional experiment to validate that. And also, as we said, the task is a finite related design. It's quite difficult to use a Leo specific contrast. So some time interval here is like 3 seconds, 1 second, even you use a Leo specific contrast. It's very difficult to separate it.
I mean, if you didn't scan the whole brain, but just a limited area, then you could use an even shorter TR with the other techniques. Is that right? Or maybe
I see that if, for example, for the even the shortest like a facial, you need like 4 seconds like that, and but it's I'm not sure how reliable you could separate some effects like event related effects if you have a time interval like 2 seconds, 3 seconds. I'm not sure how reliable you could separate that.
Okay, thanks.
So you, sorry, I just had a question. It would have been interesting to try to separate the pre-choice evaluation from the choice itself. The idea would be to, sort of draw out the time in which they're trying to make an evaluation and then have a separate time for the choice, just to sort of, I mean, maybe I missed that. Yeah, maybe.
I think I understand your question. Yeah, so your question is to separate this choice stick evaluation from the action choice selection, right? So,
Right, because it seems like the decision is made, there's a decision making, there's a evaluation period, and then the decision's made.
Yes.
It seems like there's two separate processes there.
Yes. Ideally, it will be better to separate those choice evaluation from choice selection.
Right.
But during our task design, we ask because for those super high stake decision, we wanted to induce some urgency. So urgency, some rapid choice commitment. So we ask the subject to do the selection as soon as possible. So in this way, the time interval is very short and varies across subjects, so we cannot really separate. If we want to separate, we have to design a task differently. Yeah.
And did you look at the layer effect of the salience network?
So here we only look at the layers in prefrontal. We didn't look into the layer effect in those networks. We didn't do that.
Okay, because might be interesting also to further
definitely, yeah.
Mechanism, but cool. Yeah.
Javi, you have your hand raised.
Yeah, I was going to ask something similar to what Peter asked. So maybe I'll just make a comment instead. I think, this sort of, in our experience, sort of the annoying dural veins and the upper layer veins tend to disappear when you do differential tasks. So I think, the normalization that you've used is probably not unreasonable, Yuhui.
Yeah.
No matter what you do, these sort of bigger vessels always seem to activate. And so if you have, two different sort of tasks, then, it works pretty well, I would say. If you can't use vaso or something like that, then that's a pretty good strategy, I would think. Atina asked the question that, seems to hound us whenever we submit anything layer related, which is, okay, show me the same subject again and again and again and again. So that's always, a challenge, I think. And that's very hard with your task because these higher order cognitive tasks have adaptations and learning and things like that. So it's not so easy, but it is kind of essential, I think, to do something like that, to convince, neuroscience reviewers particularly that this is real. Obviously you did, because you have the paper coming out. But in general, this always seems to be, I don't know, Renzo may have similar experiences, but this is the thing. Renzo, just since I'm thinking about this, and you asked this earlier, you will be getting a coil from us. You may not know about this. MGH will be getting a coil from us. It has 50% higher SNR all the way down into C2 compared to the Nova coil. It was originally built for John as part of a Rayner cerebellum project. Of course, John moved, but the coil is not moving with him. It's coming to MGH still. So you would actually be able to look in those areas with considerably higher SNR than what the Nova coil can do. So, that might be interesting for you.
Yeah, I agree. There's a lot of room for improvements with better coils. Specifically, yes, the Nova coil, it's really not really a cortex coil. It's just the upper cortex coil. It's not.
Yeah, it's an upper cortex coil. I mean, this coil is better everywhere. It's not just better in the cortex. It's also more efficient by about 20% transmit efficiency. I don't think that's probably too important for this.
Bobby, I don't know if you're aware, but MGH, like Stanford, have basically a knockoff of the next-gen 7T scanner. Everything is nearly the same, except they have 64-channel receiver. The Nova is a 32-channel, and we've already shown at least 50% higher SNR in these regions of the brain.
So this is 32 compared to 32. If you do 64.
Definitely. We also have a coil.
No evidence. So John's coil is quite good.
Yeah, and also I guess this is relevant. But I mean, the experiment. This is a side, but yeah, it's difficult to get the signal to noise in these locus coeruleus regions and elsewhere, so it's relevant. But also we've worked on and now have working well the transceiver approach, where we are adding the transmit coil loops, so we get 2 layers, and that puts it up to 72 channels. And there we're definitely getting higher signal to noise in these deeper regions. We're about to submit a paper, and it'll be out in a preprint.
I'm not surprised by that, actually. Even though they have overlapping fields of view. We published a paper many years ago now in, I don't even remember where it was, Neuroimage or JMR, 2 very different journals. But anyways, which showed that even with concentric coils, which have highly overlapping fields of view, when you're in the thermal noise regime, you have heat, which you are, you have massive benefits even from coils that have substantial overlap, because of course the thermal noise is independent. But if you're in the physiologic noise regime, then you get the same noise from both coils, and it doesn't help you as much. So we published this ages ago, and nobody ever seemed to have followed that up. But that, I think, would totally explain the results you're talking about.
Peter, you have your hand open.
Yeah, just really quickly, one boring question and one more potentially interesting, maybe, suggestion. Boring question is, with any of these tasks, people always ask, for 30 years, do you control for attention effects? Yes, if the stakes are higher, then you pay more attention, and that could be like a global effect that could be modulating a lot of the things that you see. The other suggestion, though, is it seems that you're throwing away a lot of potential signal by just sort of binning and looking at the average signal, trying to look at the sort of seed-based layer correlation over time. You have a short enough TR, you could actually potentially look at the correlation structure over time too. That would pull out potentially some interesting dynamics.
Yeah. So Should I? Okay. So yeah, for the attention, definitely like here, like for different stake, they will induce a different attention, and we as analysis we did like we so like, I would say it's related to the arousal, so those kind of attention, right? Those attention definitely changes, yeah, and I think Peter's suggestion about to look at those kind of dynamic, for example, correlation between LC and different networks will be definitely a very good strategy to monitor those kind of dynamic change, maybe of attention or any kind of effect to modulate those network. Yeah, I totally agree. Thank you.
All right, thanks. Then we are at the hour. Thanks, everybody. Thanks, Yuhui. And in 2 weeks, we are going to meet again. And I think that time it will be Daniel Hennel talking about veins and how to remove them. Thanks.
Yuhui, great work.
Okay.
Terrific. Yep.